Hey guys, welcome to my YouTube channel Wonderful Biology. Today I am going to talk about Robert Koch and his contribution in medical microbiology. But before that, please like, share and comment in my YouTube channel and don't forget to subscribe it. Okay friends, let us go through our subject matter, Koch Postulates, and about a great German scientist, physician, Robert Koch. Okay, look at this slide. So, Robert Koch is known as father of bacteriology. He's also known as father of clinical, he is known as father of medical microbiology because he was the first person to prove germ theory of disease. That means he is the first person to prove that bacteria is the source of infection, causative agent of infection. So he is respected as father of bacteriology or medical microbiology. So let me give some informations about this German physician. His birth was in December 11, 1843 in Germany. He died in May 27, 1910 at the age of 66 at Baden-Baden, Germany. And the subject of study of this great scientist was bacteria. Mainly, he had studied about cholera, tuberculosis, anthrax, and mycobacterium. And I have taken this information from Britannica.com. You can see that he was German physician. Let me highlight here. He is a founder, one of the founder of bacteriology. Because of this reason, he is known as father of bacteriology, father of medical microbiology. Okay, medical microbiology. Louis Pasteur is known as father of microbiology, but Robert Koch is known as father of medical microbiology. There is the difference. Okay, you have to remember this point. Robert Koch is a physician of German who is known as, once again, okay, who is known as father of bacteriology, father of medical microbiology. But Louis Pasteur, who is a French chemist, he is known as father of microbiology. You have to note this point. Louis Pasteur was a scientist who proposed germ theory of disease. But this scientist, Robert Koch, physician, he proved practically with experiments the germ theory of disease. This is the difference. Now, Robert Koch discovered the anthrax disease cycle in 1876, the bacteria responsible for tuberculosis in 1882, and cholera in 1883. For his discoveries, he was given Nobel Prize, but this Nobel Prize was for physiology or medicine in 1905, since he has discovered tuberculosis related bacteria. Okay, so tuberculosis is a disease that is caused by Mycobacterium tuberculosis. So for his discoveries in regard to tuberculosis, he received the Nobel Prize. And this is the information that we can see in www.britannica.com. So this is genuine information. Now, the very important postulates he had given, and still these postulates are in use. We follow these postulates in microbiology laboratory. These postulates are given by this great scientist when he was studying about anthrax, and he did a series of experiments. So we'll discuss about that experiment later on, but before that, let me tell you about his postulates that are mentioned here. Okay, I'll note those postulates. The microorganism must be identified in all individuals affected by the disease, but not in healthy individuals. Okay, note this point. Microorganisms must be identified in all people affected by the disease, but not in healthy individuals. The microorganisms can be isolated from the diseased individual and grown in culture. The next point. Another third point is, when introduced into a healthy individual, the cultured microorganisms should cause disease. Another point is, the microorganisms must then be re-isolated from the experimental host and found to be identical to the original microorganisms. So these are the points that are given by Robert Koch after doing series of experiments when he was studying about the anthrax bacilli, the causative agent of anthrax disease. To understand it better, let us enter in next slide. The same thing is written here, so I will not talk about this portion. You can see Robert Koch, okay, working in his laboratory. Okay, this is a illustrative figure from which I can make you understand about Koch postulates. Okay, let us focus on this illustrative diagram. Now you can see two rats. One rat is diseased, one rat is healthy. So according to the first statement that is given by Robert Koch, the animal that is diseased, the animal suffering from any disease, should have pathogen, organism, microorganisms, according to him. Okay, but those animals which is healthy, they do not have any microorganisms. That healthy animal do not have any pathogenic microorganisms in its body, according to Robert Koch first statement. So when we take out the sample from one healthy animal and another diseased animal, then look here, we can isolate the organism, microorganism, okay, in pure culture in microbiology lab. So these are the suspected pathogens that should be present in diseased animal. You can see here the bacilli, This round one is, this is RBC, okay, red blood cell, that is taken out from this animal, and this is also the red blood cell that is taken out from this healthy animal. When this sample is taken out and they are kept in culture media, incubated in optimum temperature for certain time period, then we can isolate and obtain pure culture of microorganisms. You can see the white portion. These are the microorganisms that are grown in this artificial culture medium, but you cannot see any, let me highlight here, you cannot see any microorganisms in this healthy animals, healthy animal blood sample. But you can isolate, you can obtain microorganism in pure form. There is no other microorganism present except that particular microorganism. That is a pure culture. Only one type of colony. Now, these 2 points are proved. Let us move on the third point. Now, when this white part, this white portion contains the microorganisms, the bacilli, this one, when this pathogen is now injected in this healthy rat, this is the rat, this was healthy at first, but when this pathogen, this microorganism is injected in this rat, then this rat also died here. It was killed when the pathogen was injected in its body. So it means that this pathogen which was isolated from diseased rat, when injected in this rat, then the healthy rat also got infection and it was diseased. Finally, when the sample from this rat is taken out and grown in same culture media, you can obtain the same white part, yeah, same pathogen, the microorganism that was obtained from disease animal. When cultured in lab, we can find out the pathogen, and when this pathogen was again injected in this healthy animal, this also got disease. And when the sample was taken out for the second time, we again can isolate same type of microorganism here. From this, it was clear that the cultured microorganism, when injected in the healthy susceptible host, then it also got disease. This culture was able to cause the same disease. And again, when the sample was collected from the host that was killed by the introduction of the cultured pathogen, same pathogen was isolated. This means the microorganism must be re-isolated. It can be re-isolated again from the incubated disease experimental host and identified as being identical. It means this was identical with this microorganism. So the postulates was proved in this way. First, pathogen was isolated here in culture media. It means at first he told that if this was diseased, this animal was suffering from any disease, then it should have pathogen. So he collected the sample and cultured in lab. He got the pathogen, but he didn't got the pathogen when he looked at the sample collected from the healthy animal. Now pathogen was here, the pathogen was cultured. Again, when the pathogen was injected in the healthy animal, same type of disease was caused. And when the sample was again taken from this animal which was killed by the culture pathogen, same type of pathogen was re-isolated. Hence the germ theory was proved in this way. Now let us move on next slide. There is also the same case. You can understand the same thing from this diagram too. Let us move in another slide. Now, when Robert Koch gave his postulates, then the postulates was accepted all over the world. It was a great contribution in the field of medical microbiology. But as the time passed, new and new findings were made in the field of microbiology, and there were some exceptional cases that was not followed by different microorganisms and other different diseases. Okay, even though Koch postulates was a great contribution in the field of medical microbiology, the postulates also have some limitations. The limitations are mentioned below. You can see in this slide. The limitations are that all microorganisms cannot be cultured. They cannot be isolated in artificial culture media. Okay, there are types of viruses that cannot be cultured in bacterial culture media, that cannot be isolated in artificial culture media. It requires living cells, host, for its replication. So microorganisms like Mycobacterium leprae, Treponema pallidum, Chlamydia, Rickettsia do not follow the postulates, do not satisfy the postulates of Robert Koch. We cannot grow Mycobacterium leprae in artificial culture media. We cannot isolate it. So, according to Robert Koch, we can isolate bacteria in artificial culture media, but we cannot isolate Treponema pallidum, the causative agent of syphilis, in artificial culture media. Nor we can isolate Mycobacterium leprae. Okay, we cannot isolate Mycobacterium leprae in artificial culture media. Also, there are many people who carry the disease, who carry the pathogen, but they are asymptomatic. That pathogen cannot cause disease to those persons. They are the carriers that can transmit the disease, but they themselves are not. His postulates cannot satisfy epidemiological elements of causation, immunological elements of causation, Prevention of disease by elimination of putative cause as element of causation. There are the diseases that are caused by multiple factors, not only by the pathogen themselves. One syndrome has different causes in different settings. Reactivation of latent agents can cause disease also. That postulates cannot satisfy, cannot cover immunological processes as a cause of diseases also. Immunological processes. Okay, hypersensitivity reaction is also a type of disease. Overstimulation of immune system can cause different types of diseases, not only by the pathogen. So there are also limitations of Robert Koch postulates, but we cannot ignore his postulates, because even today those postulates are very important in microbiology laboratory to find the relation between the germ and disease. For example, when you are suffering from the infection, when you are suffering from typhoid fever, then you can go in microbiology lab and have a blood culture, can prove there is the presence of salmonella typhi. It means when you are suffering from salmonella typhi, then you suffer from typhoid fever. And if you go in microbiology lab and have a blood culture, in microbiology lab, microbiologist can isolate salmonella typhi. When you suffer from tuberculosis, we can isolate mycobacterium tuberculosis in LJ medium. So there is the relation between pathogen and diseases, and his postulates are used in microbiology laboratory to diagnose different types of diseases. Now you can see that even after giving his postulates, he himself abandoned the universality requirement of first postulates altogether when he discovered asymptomatic career in cholera. Okay, asymptomatic in cholera. Now and later of typhoid fever. Asymptomatic or subclinical infection carriers are now known to be a common features of many infections, especially viral diseases such as polio, herpes simplex, HIV, AIDS, hepatitis C. As a specific example, all doctors and virologists agree that polio causes paralysis in just a few infected subjects, and the success of the polio vaccine in preventing disease supports the conviction that the polio virus is the causative agent. So there are many things that cannot be explained, that cannot be covered by Koch postulates. So there are also the limitations. And what are the major contributions of Robert Koch? So you can see the contribution that the Robert Koch made in the field of microbiology. He investigated the anthrax disease cycle in 1876. He studied about tuberculosis in 1882 and cholera in 1883. He discovered bacteria such as anthrax bacilli, tubercle bacilli, okay, mycobacterium tuberculosis, cholera bacilli. He observed the phenomenon of acquired immunity. He introduced solid media for culture of bacteria. He introduced gelatin as a solidifying agent, but on the suggestion of one of his associates that actually brought the information from his wife. He said, okay, he introduced agar as a solidifying agent. He introduced that. Before that, he has introduced gelatin as a solidifying agent. But one of his associates, when told Robert Koch that agar can solidify the culture media, He then introduced that agar as a solidifying agent. So he is a pioneer person to use agar as a base for culture media. He also developed pour plate method and was the first to use solid culture media. Was also developed media suitable for growth of bacteria isolated from the body use as nutrient sources. The result was the development of nutrient broth and nutrient agar media that are still in wide use today. He introduced methods for isolation of bacteria in pure culture. Now, pure culture. This is a great contribution. He described hanging drop method of testing motility. He introduced staining technique by using aniline dye. He invented hot air oven, steam sterilizer, and introduced methods to find out the efficacy of antiseptics. So the main contribution are listed in this slide. You can go through it. And then this is information that I have taken from Sakja. The book every student love to read. Same thing is written. So you can go through these portions also. Robot Koch is a very important person. Many university ask the questions related with Robot Koch and his postulates in their exams. I hope you will now able to describe about the contribution and the importance of Koch postulates in microbiology. In next slide, I will explain about the contribution of Louis Pasteur. Before leaving, I want to mention you one thing that scientists have claimed that they had found the way to cultivate. They have found the way to culture Treponema pallidum in artificial culture medium in petri dish, according to drugtargetreview.com, but it is still difficult to culture Treponema pallidum, and hence this Treponema pallidum is still controversial spirochete that had challenged Robert Koch's postulates. This much for today. Stay safe, stay healthy from COVID-19. Bye bye. Thank you.